Welcome, everyone, to this foundational lecture in immunology. Today, we are going to explore two of the most critical concepts in the entire field, antigenicity and immunogenicity. For anyone studying zoology or medicine, understanding the precise difference between these two terms is absolutely essential. It is the secret behind how our immune system works, how we design life-saving vaccines, and why some people develop severe allergies. By the end of this video, you will not only know the biological definitions, but you will also be able to calculate and prove these concepts mathematically. Let us dive right in.
To begin, let us build our intuition using a simple analogy. Imagine a highly advanced security system installed in a large bank building. If an intruder breaks in, the motion sensors detect them, and the main alarm goes off, alerting the police. In biology, the ability of a substance to trigger that main alarm is what we call immunogenicity. Now, imagine the security guards already have a mugshot of the intruder. When they see the intruder in the hallway, they recognize and tackle them. That ability to be recognized is antigenicity. Sometimes a tiny piece of dust floats by. It does not trigger the main alarm, but a guard looking closely can still recognize it. This shows us that starting an alarm and being recognized are two completely different things. Now that we have our analogy, let us look at the formal scientific definitions, starting with immunogenicity. Why does this matter? Because without immunogenicity, our bodies would be completely defenseless against new infections. Immunogenicity is defined as the intrinsic ability of a substance to induce a specific immune response. We call a substance that can do this an immunogen. When an immunogen enters the body, it sounds the biological alarm. This alarm can trigger a humoral response, which involves B cells producing antibodies, or a cell-mediated response, which involves T cells actively hunting down infected cells. The key takeaway here is the word induce. An immunogen is an active initiator of the immune defense process. Moving on from the initiation phase, we must understand what happens after the immune system has deployed its weapons. This brings us to antigenicity. Antigenicity is the ability of a substance to bind specifically to the final products of an immune response. These products are typically secreted antibodies floating in your blood or specific surface receptors on T cells. Notice the crucial difference here. Antigenicity is purely about binding, not triggering. Just like the dust in our bank analogy that the guard can see but does not set off the main alarm, a highly antigenic substance can be perfectly recognized and grabbed by an antibody, even if it never had the power to wake up the immune system in the first place. This distinction leads us to one of the most important rules in immunology. Because an immunogen can trigger a full response, it will certainly be able to bind to the antibodies produced by that response. Therefore, all immunogens are antigens. However, the reverse is not true. Not all antigens are immunogens. A perfect example of this is a class of small molecules called haptens. Haptens are antigenic. They can easily bind to existing antibodies. But they are too small and simple to trigger an immune response on their own. They are completely ignored by the body's alarm system unless they physically attach themselves to a larger carrier protein. Once attached, the entire complex becomes visible and triggers a massive response. Before we look at the mathematics, let us understand why this specific distinction matters so much in modern medicine. It is the absolute foundation of vaccine design and allergy treatment. When scientists create a vaccine, they must ensure the substance is highly immunogenic. If a vaccine component is only antigenic, your body will completely ignore it, and the vaccine will fail to train your immune system. Conversely, understanding haptens is crucial for treating allergies. Small molecules like penicillin are haptens. Normally, they are harmless and not immunogenic. But in some people, penicillin binds to larger proteins in the blood. Suddenly, this new complex becomes highly immunogenic, triggering a severe, life-threatening allergic reaction. This is why manipulating these properties is so vital. Now that we understand the biological concepts, let us look at how these physical properties combine mathematically. While immunology is a biological science, we can define a theoretical immunogenicity score to see exactly how different factors interact. The formula is I equals K times the product of F, M, C, and D. Here, I is the overall immunogenicity score. The variable K is a biological constant, depending on the host's genetics and immune health. Inside the parentheses, F represents foreignness, or how different the molecule is from the host. M stands for molecular weight, as larger molecules trigger stronger responses. C is chemical complexity, and D represents degradability, which is how easily immune cells can process the antigen. If any of these intrinsic variables, like foreignness or molecular weight, drop to near zero, the overall score I drops significantly, and the immune system ignores the molecule. Let us apply this formula to a concrete example. A scientist is evaluating a newly discovered bacterial protein for a potential vaccine. We are given the host biological constant K is 1.2. The protein has a foreignness score F of 8, a molecular weight factor M of 10, a chemical complexity C of 5, and a degradability D of 4. We need to calculate the total immunogenicity score. We start by substituting our given values into the formula. Following the order of operations, we first multiply the intrinsic properties inside the parentheses. 8 times 10 times 5 times 4 gives us 1600. This is the raw potential of the molecule. Finally, we multiply by the host constant, 1.2, giving us a final score of 1920. Because this is a very high score, it physically means the bacterial protein will provoke a robust immune response, making it an excellent vaccine candidate. Now that we have successfully used the formula, let us use it to mathematically prove the concept of haptens and carrier proteins we discussed earlier. We want to prove why haptens cannot trigger a response alone. We start by evaluating a small, isolated hapten floating in the bloodstream. The host constant K is 1.0. The hapten is highly foreign, so F is 9, but its molecular weight, M, is only 0.5, and its degradability, D, is just 1. Its complexity, C, is 2. Substituting these into our equation, we multiply 9 by 0.5 by 2 by 1, which gives an intrinsic value of 9. Multiplying by the host constant leaves us with a score of 9. This score is extremely low and practically negligible. It mathematically proves that the isolated hapten is absolutely not immunogenic and cannot trigger the alarm bells. This extremely low score brings us to the next step of our proof. What happens when scientists attach this exact same hapten to a large carrier protein? The core hapten structure is still present, so the foreignness, F, remains 9, and the complexity, C, remains 2. The host constant, K, is still 1.0. However, the addition of the massive carrier protein drastically increases the molecular weight factor M to 15, and the degradability D increases to 8. We now substitute these updated values into the exact same equation to evaluate the new hapten carrier conjugate. We are setting up the multiplication of 9 times 15 times 2 times 8. This step shows how manipulating just two physical properties, size and degradability, can fundamentally alter how the immune system perceives a threat.
Let us complete the calculation to see the final result of our proof. Multiplying the new intrinsic values together, 9 times 15 times 2 times 8, gives us a massive raw potential of 2,160. Multiplying by our host constant of 1.0, the final immunogenicity score for the conjugate is 2,160. Compare this to the mere 9 we calculated for the isolated hapten. The score has dramatically jumped. This mathematically demonstrates that attaching a small, non-immunogenic hapten to a large carrier protein makes the entire complex highly immunogenic. The immune system will now mount a massive defense that also targets the small hapten. This exact mathematical principle is how conjugate vaccines, like the one for Haemophilus influenzae type B, are successfully engineered to protect infants.
So, we have proven the carrier effect mathematically. Now, let us visualize these relationships clearly with this diagram. What you are looking at is a large outer circle representing all antigens. These are all the substances that can bind to immune products. Completely inside this is a smaller circle representing immunogens, the substances that actually trigger the immune response. Notice the blue stars inside the inner circle? These represent large molecules, like proteins, that are both antigenic and immunogenic. But look at the space between the two circles. Here we see small red dots labeled haptens. Because they are inside the large circle, they are antigens. But because they are outside the inner circle, they are not immunogens. This diagram perfectly captures the golden rule we discussed earlier. To wrap up our lesson, let us review the most important ideas we have covered today. First, always remember the core distinction. Immunogenicity is the active ability to start an immune response, while antigenicity is simply the passive ability to be recognized by the products of that response. Second, keep the golden rule in mind. All immunogens are antigens, but small molecules like haptens prove that not all antigens are immunogens. Finally, we saw mathematically how specific physical factors like foreignness, molecular size, and chemical complexity drive immunogenicity. By understanding and manipulating these factors, scientists can create life-saving vaccines and treat severe allergic reactions. You now have a complete foundational mastery of how the immune system decides what to attack and what to ignore. Great job!